Hello everyone and welcome back to Anatomy 5. Today we're going to be talking about Lab 12, which covers the eye. Now there's going to be a few purposes of this lab. One is to talk about the anatomical layout, so the structural organization of the eye and the eye accessory organs, and then also the function. So how does the eye accomplish the main function, which is vision, allowing us to basically interpret light stimuli from the environment. So what we're going to do is we're going to do a brief overview of the visual system, talk about some of the key components, and then we're going to switch to a PowerPoint slide where we review and then also do a few sketches. And then lastly, we'll follow that up by talking about some of the activities that you're going to do, which will test the physiology of the visual system. So let's go ahead and get started. Now, first off, think about what the main purpose is of the visual system. The main organ are going to be the eyes. And so the eyes are going to basically be the window to the soul, you could think of them. They're going to have the machinery to interpret the light, turn it into neural signals to send to the visual processing areas of the brain. So we're going to focus mostly on the eyes. And so the eyes are surrounded by bones of the orbit. And we covered these when we discussed the skeletal system. And that's essential for protecting these vital organs. Now, one of the accessory muscles we're going to talk about are called the extrinsic eye muscles. And we have them right here. And I'm going to go ahead and label them now. So when we talk about them a little bit later, it'll make some more sense. So right now we are hiding the rest of the muscles. So these extrinsic eye muscles are very important for allowing us to control eye movement. So we have the lateral rectus, we have the medial rectus, we have the superior rectus, we have the inferior rectus, we have the inferior oblique, and then we have the superior oblique. Now again, we're going to come back to these, but I just want to talk generally about this. So first off, I want to just ask you if these are voluntary muscles. Can we voluntarily control them or consciously control them? And the answer is yes. These are skeletal muscles. And if you had to think about that question, just point your eyes to the ceiling and think about it. Well, that gives you an example. So our eyes, it's very essential that our eyes make very precise movements. And these movements could be to the side in the lateral direction. They could be down, they could be up, they could be even at angles, and we can even do circular sort of rotations. So all of these complex eye movements are going to be produced by the contraction and activation of either one of these muscles or a combination of them. Now, I'll just give you an idea. Let me go ahead and erase this, and we're going to come back to these. I'll give you an idea of what happens when you contract these muscles. So for instance, this superior rectus muscle, if you contract that, its insertion is on the sclera. So if you contract this, it's going to cause the eye to rotate in an upward direction. Now, on the other hand, if you contract the inferior rectus, it's going to cause the eye to rotate in the downward direction. And I say rotate because remember, the eye is a sphere, and so when it moves downward, it's going to still be in a rotating movement. Now, this lateral rectus is going to be involved with abduction of the eye, so moving the eye away from the midline or in the lateral direction. And then the medial rectus is going to be involved with adduction. And so that's going to be movements towards the midline or towards the medial part of your visual field. The superior and inferior obliques are a little bit more difficult because the movements are sort of counterintuitive. Let's start with this inferior oblique. So when the inferior oblique contracts, it actually is going to cause the eyes to move up and out. So a superior lateral rotation. So again, the inferior obliques, when those contract, they're going to cause a superior lateral movement. So up and sort of out to the lateral direction. The superior oblique is going to do the opposite. If the superior oblique is activated, it's going to cause a downward and a downward in lateral movement. So basically it's the downward and being able to go back and forth. So remember when we learned about other muscles called obliques, the external obliques and the abs? And in lab, we talked about how having muscles that are arranged in the fibers are arranged in different directions, allow for different movement. So you imagine if you just had the superior, inferior, lateral, medial rectus, you'd really be able to do movements left and right, up and down, all right? And so these obliques are going to allow for rotation movements. So not only up and down, but basically to be able to rotate in all these different directions. And if you think about the advantage of this, if you had to look at something really quick, you don't have to move your whole head or your whole body. You can just simply quickly make an eye movement towards that particular region of space. So again, we'll come back to these in the PowerPoint slide, but I wanted to give you an overview of what these extraocular muscles are doing. Now, let's talk about the eye. So here we have the eyeball. And before we go into some of the more detailed structures, let's talk about some things that you may have, come into this class already knowing some features of. So you're probably not used to seeing the eye out of its socket like this. So right now we're looking at an anterior view. Now, we kind of flip it to the side a little bit. This transparent portion on the front, this is the cornea. You may have heard of this structure. The cornea is transparent, and that's super important because you need to get light rays into the eye. Now, the cornea gives rise to the white part of the eye, or the sclera, that surrounds the eyeball. And the sclera is what those extraocular muscles are going to be attached to and produce those eye movements that we just discussed. So posterior or behind that cornea is the lens. The lens is this very highly elastic structure, and that's going to be super, super important to basically focus and refract incoming light from the world back onto that retina portion that's going to be able to transduce the light stimuli basically into a nerve signal so our brains can perceive it. So we're going to talk about that lens. Now let's go back here. Behind the cornea is the iris. This is the portion of the eye that has high variation in terms of its color. Some people have green and blue irises. Some are hazel or very darkly pigmented. And we'll talk about the iris. It's very important. It's composed of several types of muscles, and the iris is going to be essential for controlling the diameter of the pupil. So the pupil, we have kind of shade this in here. The pupil you can think of as the aperture. It's going to allow or control the amount of light that comes into the eyes by either contracting and shrinking and letting less light in, or dilating and expanding and letting more light in. So the size of the pupil is going to be controlled by the muscles of the iris. Now, once incoming light comes through that cornea and then is focused and refracted by that lens, that light is going to be sent to this neural layer, or the retina. And that's what's highlighted here in green. And so the retina is going to line the inner membrane of the eye, and that contains the photoreceptor cells, the rods and cones, which are going to be essential for sensing the light and then transmitting that information via the optic nerve or cranial nerve 2 to the visual processing areas of the brain or the occipital lobe. So that gives you a little idea of some of the main components of the eye and hopefully some of those structures you were familiar with. Now let's go into a little bit more detail about these layers. The eye you could break up into three, actually I'm write it over here, 3 layers. And we're going to describe these from the outermost layers going in deep, or the superficial layers to deep. So the outer 2 layers are called the fibrous. The fibrous coat. And the fibrous coat is going to make up the sclera and the cornea. So that transparent portion that's going to allow light to come in, the cornea, which gives rise to the sclera, the white part of the eye that surrounds it. The next layer that's deep to the fibrous coat is going to be the uvea. And the uvea is composed of the choroid, the ciliary body, and the iris. Now, we've talked about the iris so far. The iris is the colored portion of the eye, and it's important for controlling the diameter of the pupil. Now, the choroid, what's highlighted here, if you were to section an eyeball in a coronal section and then look at these different layers, the choroid is going to be a pretty darkly pigmented layer, and that's actually a functionally important thing because it's going to basically enhance the photoreceptive layer or the retina. Now you'll also see the structure called the ciliary body. So that's what's highlighted right here, the ciliary body. Now the ciliary body, I'm going to kind of go into this inside view, is going to be essential for controlling the size of the lens. So we're going to talk about this a little later when we discuss near point vision or accommodation. But you can see that the ciliary body is connected to the lens via these structures called zonule fibers or accessory ligaments. And those accessory ligaments are going to either allow the lens to be flat and focus on close-up objects, or to basically straighten up and focus on distant objects. And so we'll talk about that process when we get to the PowerPoint slides. But I want you to see those here, so when we discuss the ciliary body and then the choroid, which the ciliary body gives rise to the choroid, you'll have basically seen it here. And then lastly, we have the neural layer or the retina, and that's what contains the photoreceptive cells, the rods and the cones. Now, I also mentioned that there's accessory organs, and we discussed the extraocular muscles, which we're going to come back to. But let's also talk about the other accessory portion of the eye, and that's going to be collectively called the lacrimal apparatus. Now, the lacrimal apparatus contains the lacrimal glands. That's what I showed you right here. That's the lacrimal glands. And these are going to produce lacrimal fluid, or more commonly known as tears. And so these lacrimal glands are going to be producing tears, and they're going to be excreting the tears into basically on the surface of our eye via these structures called excretory ducts. And so it's hard to see, so I'm going to zoom in. So we have the gland here, and then these excretory ducts. Okay, so if you remember the difference between endocrine gland and exocrine glands, these are going to excrete these tears onto the surface of our eye. So if we zoom out, just move this here. Okay, so we zoom out, the glands are going to produce tears The tears are going to be released from these excretory ducts, and then they're going to travel across the surface of the eye to lubricate it and protect it. And then these tears are going to drain into these structures called lacrimal canaliculi or canaliculi. And so the tears drain into these canals, these really thin canals. We have one on the lower and upper portion of our eyes. And then finally, they drain into the lacrimal sac and then empty into the nasal cavity. So if you ever experience a moment where you've been crying a lot, you might notice that you get a runny nose along with that, and that's part of the tears draining into the nasal cavity. So this process of forming the lacrimal fluid or tears in the glands, excreting them, and having them pass along the surface of the eye is going to be really essential for protecting the eye from foreign objects coming in and basically maintaining the health. Now, the eye interior is, you can think of it as the window to the function of the eye, right? So the eye's main function is to take in information from the world, visual information, sense that with those receptors, and then transduce that or transmit that information to the visual processing areas. So let's start with the lens. I said that that's the elastic structure that's going to basically change size and shape to focus and refract light onto the retina. Now, behind or posterior to the lens is what's called the vitreous body. That's going to be filled with a aqueous fluid called the vitreous humor, which is composed of mostly water, but it's also going to be important to basically keep pressure in the eye to hold that retina against the back of the eye. And of course, we have the retina, which is the photoreceptive layer. Now, speaking of the retina, so I have the retina highlighted here. Now, we're going to talk about some important anatomical and structural items here that have physiological consequences. So let me discuss this and talk both about the anatomy, the organization, as well as the function. So the retina, as I was saying, is going to be transmitting light to the brain, to the visual processing areas, and it does that via cranial nerve 2 or the optic nerve. So this retina is going to be taking in light information from the entire retinal layer and transmitting this information via the optic nerve to the brain. Now, if you look at the back of the retina, so we have an eye, and if it's sectioned in layers, so it's a mid-sagittal section, so we can look inside. If you look at the back of the retina, there's a couple important landmarks. So first off, I'm going to highlight the optic disc. So the optic disc, which is highlighted here, there's a couple important things that are going to be, you could think of them as exiting the eye at this point. One is going to be the optic nerve, so cranial nerve 2, that's going to be bringing visual information from the retina to the visual cortex. And then also you have these central retinal arteries and veins. So this is going to be bringing blood to the eyes and bringing blood out of the eyes. And so those also enter and exit via that optic disc. So the consequence of that is that at the optic disc, there are no photoreceptive cells, no rods and cones that can transmit that information. So it's also known as the blind spot. And you'll be exploring the physiological consequences of that in one of the activities in this lab. Now, another thing I want to point out, it's not shown here, but we could get a different color, and over here, I'm going to draw something on the back of the retina, and this is just for illustrative purposes, but it's called the macula or the fovea. So let me zoom out a little. It's going to be right here. So this is the macula. You may have also heard it called the fovea centralis. Okay, so let's have the eye like this. Let's say that we have an object out in the world, maybe it's a tree, okay? And we're focusing on that tree, so we're looking at that tree. Light's going to be coming in, refracted and focused by that lens. And if we're looking straight at it, the light's going to be directed towards that fovea. So the fovea or macula has the highest density of rods and cones, the photoreceptor cells. So we have our highest visual acuity at that region. Now imagine if you're looking at a tree and, maybe this is going to be a bad drawing, but a predator, a lion or something was in your peripheral vision, but that light was being directed to that blind spot where there's no photosensitive cells. You wouldn't perceive that. And so that's why we call it the blind spot, because in your peripheral vision, if you're looking straight at something, you're not going to sense that area of the retina. All right, so we'll talk more about that when we get to that activity. So we're gonna end this with a few videos that summarize this and talk about how we get that visual information into the brain. So I said the retina is the photosensitive layer, and what that means is this layer contains rods, which are going to be important for sensing basically brightness of light, bright and dim light, and then cones, which are essential for interpreting color. So let's go ahead and watch this, and I'm going to pause it a few times and highlight a couple important things. So first, they're just showing a person's eye. You have some incoming light waves, right? That's being transmitted to the retina. That impulse or that nerve impulse is then going to be sent to the visual cortex, so we can perceive that as a visual image. Now, we first talked about the cornea. That's the transparent or the window to the eye that's going to allow light to come in through the pupil, which is highlighted right here. And the pupil is going to change size, either dilate or constrict. Now, once light comes in through the pupil, it's going to be refracted and focused via that lens back onto the retina, where we have the photosensitive cells. And they're going to zoom in here, and I'm going to pause. And what they're showing here essentially is the retinal layer. So we have rods and cones here, right? And so when light comes in, they're going to activate these rods and cones. So they're going to basically send action potentials down this optic nerve to the visual processing centers of the brain. So those photosensitive cells are going to be activated by the light. We have the rods and the cones sensing either bright and dim light or colors. And then they send that information via the action potential down cranial nerve 2 to the occipital lobe, or the visual processing areas. Now, the last thing I'll show you, just because this video highlights this really well, one of the activities we're going to be doing in the lab is the visual acuity, all right? So this is what you would do if you go into a doctor's office or basically an optometrist to have your eyesight checked. And there's other health reasons you could do this. But we're talking about the Snellen chart. That's where you stand 20 feet away from a chart and the doctor asks you to read letters of different size. Now, let's just talk about what happens in a normal person's eye. And I say normal, meaning they have 20/20 vision without any corrective lenses or glasses. So what happens is when that light comes in, that lens is refracting and focusing that image to the correct distance right on the retina. And so you have a focused image. Okay, so this is what we mean by normal vision. Now, people like me who need contacts can have disruption of this normal vision. So one scenario is where that information is actually not making it quite to the retina. So this would be called nearsightedness or myopia. And so that image is basically in front of the retina. So you have to wear corrective lenses to get that image extended back to this retinal portion. Now, you also could have what's called farsightedness or hyperopia, and that's where the image is projected to a region beyond that retina. So, again, it's not going to be in focus. So, you can think of normal vision, that image is focused back onto the retina in the proper place, and someone with normal vision would have 20/20. And for example, someone with myopia might be without glasses be 20/80, and we'll talk about what that means. And then someone farsighted might be 20/15, and we'll talk about what that means as well. Okay, so that concludes the introduction to the visual system. And now what we're going to do is we're going to switch over to a PowerPoint, summarize a few of these key things, do a few sketches, and then talk about the activities you're going to do for the visual system. Hello everyone and welcome back. So now that we've reviewed the eye, the general anatomy, and talked some about the function, let's go over what we're going to be doing in Lab 12 specifically, which is going to cover the visual system. So the activities you're going to be doing in Lab 12 are going to start with the anatomy of the eye. Now, I'm hoping we're going to have cow eyes to dissect in person, and then if you're doing a virtual lab, we're going to have some good online activities that are going to walk you through the anatomy of the eye. So you're going to be doing a sketch that covers the anatomical organization and layout, and then I would include a table of the function of those different parts. And then you're definitely going to want to include the extraocular muscles in that, as well as the lacrimal apparatus. There's going to be a portion that's going to be reviewing or testing yourself on the eye anatomy, and then a dissection activity, whether that's in person or virtual. Now, I want to talk about these visual tests a little bit. And I have a couple slides to talk about these individually, but we're going to be testing visual acuity, the accommodation reflex or near point vision, the blind spot, and an after image. So let's go ahead and get started. And what I would suggest is getting some markers, some crayons, any sort of materials that you might have that you want to do some sketches with. All right, let me just make sure everything's set up on my end. Okay, so first off, let's review the layers. So remember I said that when we talk about the eye, we can break it up into 3 layers. And we'll talk about these going from superficial to deep, so the outermost layers to in. And start with the fibrous. The fibrous layer. And the fibrous layer is going to be composed of the cornea and sclera, the white part of the eye. Then we have the uvea, or the vascular portion, which comprises the iris, the ciliary body, and the choroid, that pigmented layer. And then lastly, we have the neural layer or the retina. All right, so let's go ahead and draw this. And what we're going to do, again, remember, this isn't anatomically correct, but the purpose of these sketches are to help you associate these different layers in your mind and help you learn the organization. So what we're going to do is we're going to start with the cornea, the transparent portion. Okay, so it doesn't have to be perfect. With that said, I'm going to restart that one. Okay, so we have the cornea. And remember, the cornea gives rise to the sclera, or that white part of the eye. And we'll leave a room for that optic disc, which we'll talk about in a bit. So we can start labeling some of these components. We have the cornea. Whoops, it's not very clear. I want to make sure this is clear for you all. The cornea, which gives rise to the sclera. All right. Now, behind or posterior to that cornea is the lens, that elastic structure that's going to be essential for refracting and focusing that light onto the retina. All right. Now let's go ahead and draw the uvea, the iris, the ciliary body, and the choroid. Now to do that, I'm going to get a different color. And I'm going to start by drawing the iris. Okay, so the iris, that colored part of the eye, which gives rise to the ciliary body. You have an iris on the upper and lower portion and a ciliary body on both portions. And then that's going to give rise to the choroid. All right, so let's go ahead and label these and outline them a little bit. So we have the iris here. The ciliary body. Remember, that was that structure that's involved with changing the shape of the lens. And then we have the choroid. And those three make up the uvea, or the vascular layer. Now, remember those suspensory ligaments, or those zonule fibers? We can go ahead and draw those in. Those are going to be connected to the ciliary body, connected to the lens. And remember, depending on, we'll talk about this with accommodation, depending on whether these zonule fibers are relaxed or contracted, it's going to change the shape of that lens. Now, we can also draw those extraocular muscles that we talked about that are going to be inserting onto the sclera, and those are essential for the various types of eye movement. So extraocular muscles. And the region right behind or posterior to the cornea, but in front of the lens, that's going to allow light in. Remember, that's the pupil. And this region in front of the pupil behind the cornea is called the aqueous humor. And that's composed of primarily a water solution that's going to be important for and transparent, right, to allow light to come in. Now, behind the lens is the vitreous humor, which also is a fluid-filled cavity. And now let's go ahead and draw the retina, the neural portion. So I'm going to do that with a bright pink color. So we have the retina, the photoreceptive layer, and then the retina, remember, gives rise to the optic nerve. So the optic nerve is going to be coming out the back. So let's go ahead and label this. We have the retina, the optic nerve, and then remember we said that there were those central retinal arteries and veins that are going to be exiting and entering via that optic disc area. So we can go ahead and draw those and label those the central artery and veins. All right. And then remember this region where the optic nerve and those central artery and veins exit and enter the eye is going to be the optic disc. And that's also known as the blind spot, which we'll talk in a bit. Now I'm also going to highlight that macula. So what we could do is just with a different color, maybe a dark green, we can highlight that macula or that fovea centralis or fovea. Remember, that's going to have that high density of photoreceptors. And so when we're focusing on an object and trying to read it, that's where we're going to have that high density of rods and cones, which allows for visual acuity. All right. Now, I believe that covers the majority of the components that we have. Let me make sure I've gone and labeled all the essential components. So I would review this, and like I said, it's probably helpful to make a table that covers the different functions of these parts. For instance, the cornea is transparent, it's going to protect the eye and allow for light to pass through. The lens is important for focusing and refracting light back onto the retina. Now, I said we're going to talk about the accessory organs, and we discussed the lacrimal apparatus previously, and we also introduced the extraocular muscles. And so I want to talk about those a little more detail right now. Sorry, I'm just trying to get my computer set up. Okay, so let's start with talking about the muscles and then their function. So what are the general movements that they're producing? And remember I said a lot of eye movements are going to be using a combination of this. So let's start with the superior rectus. So the superior rectus is shown right here. It's attached to the sclera. And so when the superior rectus contracts, it's going to elevate the eyeball or rotate it upwards. And this is innervated by cranial nerve 3, the oculomotor nerve. Now the lateral rectus, so that's going to be on the lateral sides of the eye. And this can be difficult if you just get a single eye. It might be difficult sometimes to know which is the lateral and which is the medial. But the lateral rectus, it abducts, so it's going to rotate the eye laterally or away from the midline, and that muscle is innervated by the abducens nerve or cranial nerve 6. Then we have the inferior oblique. Okay, so the inferior oblique, remember, is this muscle right here that's at an oblique angle to the eyeball. So the inferior oblique, when it contracts, it produces a movement which we'll call superior and lateral rotation. So superior and lateral. So it's going to be lateral, meaning if it was my left eye, it's going to be this way, and if it's my right eye, it's going to be this way. So to the lateral direction, or so you can think of it almost as abduction. The inferior rectus, so the inferior rectus muscle, when that contracts, it's going to rotate the eyes downward or depress the eyes. And then we have the medial rectus. So the medial rectus is going to be on the medial portion of both eyes. And when that contracts, it's going to cause adduction of the eye and bring the eyes towards the midline. And then lastly, we have the superior oblique. The superior oblique is interesting because it's connected to this structure called the trochlea. And the superior oblique, when it contracts, it causes what's called inferior and lateral rotation. So downwards and away from the midline. And it's innervated by another cranial nerve called the trochlear nerve. Okay, so just to conclude, we have these extraocular muscles that are going to be responsible for the various types of eye movement. They're innervated by different cranial nerves. So remember, those were the portions of our peripheral nervous system that are going to be oftentimes mixed nerves, having these motor nerves as well as sensory nerves coming into the brain. Now let's go ahead and talk about some of the visual activities you're going to be doing. And we're going to start with visual acuity. Now, if you look on this at this chart on the left, this is what's called a Snellen chart. This is what you're going to see if you go see an eye doctor to have your eyes checked. Now, we consider this preventative healthcare, right? We have to, have good vision to get around in the world, but it also could be done to assess eye injury or other eye conditions. I think I've shared with the class that I personally am legally blind, which occurred about 3 years ago. And so I've had a lot of experience with different types of eye exams that weren't just, a routine checkup. Now, the key with visual acuity exams is that they are careful, controlled ways of assessing someone's visual acuity. Now, how well can they see compared to the average person? And these have to be reliable and consistent. So, if someone has an eye disorder, an eye condition, you might have to do one of these visual acuity tests and track it over time. So it's important to be consistent with it. So let's talk about this Snellen chart. So if you look at this chart right here, you see these letters. So we're going to say that this is the top. And then down here we have the bottom. Whoops. Bottom. All right. So what you do is you have a patient stand 20 feet away from this chart. And it's not about 20 feet. It has to be exactly 20 feet away. And then what you do is you have them start from the top. And this would be the most basic and general way of doing this test. If you go to an eye doctor, they might immediately zip down to one of these lower rows. But if you're going to assess a disorder or an eye condition, you want to start from the top. And so that the person is standing 20 feet away and you're the observer or the clinician who is near the chart assessing the patient and making sure that they have one eye covered. So again, they're doing this test with one eye covered and the basically visual information from one eye. So you have them read the first letter, and if they can read it, you go down to the next line, you have them read it. And essentially, let's say that the person without their contacts or glasses said, okay, I have E, I got F, P, and then T. Oh, I don't know what those other two are. Maybe that's a D and an S. And you say, nope, you got those wrong. So essentially, you would go to the above line, and these are the ones they got right, F and P. So this person without glasses, you would say, is 20 over 100. And what that means is that at 20 feet, this person can see what a normal person sees at 100 feet, okay? And again, normal means someone with 20 over 20. So what would that look like? If that person had 20/20 vision and they were standing 20 feet away from this chart, they would be able to read this line down here, which is called sorry, let me erase that so you can see. It's the DefPotec, DEFPOTEC. And so that's 20/20. And that means that if a person is standing 20 feet away and can read those letters, but not the ones below, and then everything above, that person would be considered to have normal or 20/20 vision. Okay? So it gets a little bit more complicated, and let's take this for example. We have a patient who's 20 feet away. They have one eye covered. They get this line, they get this line, they get this line, and then they get this line. Then you ask them to go down here, the 20 over 40. And let's say they get a few letters. Let's say that they get all the letters but 2. So they miss this one and this one. If they only miss 2, you can account for this line, and you would say that this person would be um, 20 over 40 minus 2 because they missed 2 letters. If they miss any more than 2, then you have to go to this line above, and they'd be 20 over 50. Okay? So you have to get all of the letters right with only missing 2 to basically pass that particular line. And then remember we said that myopia or nearsightedness is anything that's basically 20/20 and above this direction. That would be myopia. And then hyperopia is 20 over 15 and below. And so that would be where images are focused beyond the retina. So that's the visual acuity chart. And so you're going to be doing this in the lab with a partner, or you'll be doing a virtual lab online that simulates this. Next, we're going to talk about visual accommodation. Now, the background to this is that the lens, being an elastic component, it focuses the light back onto the retina, and so it has to change shape depending on the distance of the object, all right? And so the bottom line is that as we age, that elasticity of the lens decreases. And so we test this accommodation or near point reflex as a way to see how well a person can focus on close objects versus far away objects. You could also think about this as, what is the minimum distance that you can hold something away from your face and focus on it? All right, and this is the near point focus distance, which decreases as we age. So if you're really young, you could probably hold something really close to your face and read it, like a book or something. Whereas if you've ever noticed elderly people, they might be holding something really far. It's because their near point is much further. So let's talk about the anatomy of this. So first off, let's just review. We had the eye, which we have gone over. And if we zoom in here, we have the lens. And then remember, we have those ciliary muscles and ciliary bodies, which are holding in those suspensory ligaments. Ciliary body, or ciliary muscles, and then the suspensory ligaments. Okay, so let's consider 2 scenarios here. First off, let's consider a scenario where, a person's focusing on something at a distance. So maybe they're outside and they're looking at a beautiful mountain scene, some trees at a distance, okay? So if you're focusing at a distance, you're going to want that lens to be pretty straight, all right? Now, in order to do that, those zonule or those suspensory ligaments have to be very taut or contracted, okay? Now, in order for that to happen, the ciliary muscles have to be relaxed. So the ciliary muscles are relaxed, and that allows for these zonule fibers to be taut or stiff. And that's going to pull that lens pretty flat and allow you to focus on those distant objects. Now, let's say that this person wants to quickly change their point of focus and maybe read a document. Let me move my head. Read a document on a paper close by. So this is the accommodation reflex. So in order to focus on something close, not only do we have to change our eyes and basically point them inwards towards this close object, that's part of the accommodation reflex, but we also have to change the shape of this lens. So essentially what we want is the lens to be more convex and rounder. So that's going to require those suspensory ligaments to relax so they're not pulling on the lens. And that's caused by contraction of the ciliary body and ciliary muscles. So contract, and then these are relaxed, and it allows that lens to be more convex to allow light to be focused at a close distance. So this is what we call the near point distance. And this near point distance increases with age. So as you age, you're going to have to hold this document or your phone further and further away from your face to be able to read it. Now the next one, the blind spot, this is pretty easy because we already talked about the anatomy, which explains this function. And so right here we have a cross section, or we're looking at the back of the retina. And so you have the macula densa, or the fovea centralis, that has that high density of photoreceptor cells, and then you have the optic disc, which is where the optic nerve enters and exits, as well as those retinal arteries and veins. And so that's what we call the blind spot. All right. And so again, when we focus on things, when we're looking at stuff, when we're reading things, or you're looking at a friend across the room, the image is focused onto that retina. That's the key. That's where we have that high density of rods and cones. We have the high visual acuity. And anything in the other surrounding regions we consider what's called the peripheral vision. Okay? So when you're looking at something straight on, you can notice things in your periphery. Well, if something happens to land on that optic disc that's in your peripheral vision, it's not going to be detected because there's no rods and cones in that region, so it's going to not appear in your visual field. So to test this, you have basically a piece of paper and you focus on a dot, and in the peripheral vision on the paper there's another dot, and as you move the paper away, this dot in their peripheral vision eventually is going to hit that blind spot, and you're going to see it disappear from your perception. And that's an example of the blind spot in action. Now, the last one I'm going to talk about, I'm going to just give you a brief explanation because this is without a doubt the most complicated one to really understand. And so what we're going to say is, well, this is the after image. All right, so essentially what you're going to be doing in this lab is you'll be staring at colored papers, or you could stare at this thing, which has a bright green background with red text. So you stare at this image or a colored paper or an American flag, something like that, for like 30 seconds to a minute. And you stare at it really intensely, focusing on the center without moving your gaze. Try not to blink. Then you quickly remove this image or the color and put a white piece of paper, and you blink a few times, and what you see is what's called an after image. And the idea is that when we okay, so let's say we're looking at something. So here's our eye, some light's coming in. Those photoreceptor cells, what happens is they become activated so much by these particular colors, so those cones, that over time they stop firing. All right. And if this was green and this is red, the opposite colors, those cones are suppressed. Okay. And so when you remove that image, the ones that were suppressed start firing. And so in a sense, it's an optical illusion. And so you see the opposing colors. So let's say you had a red square, you looked at it for 30 seconds, and then you put a white piece of paper there, it's going to appear green, all right, because those are the opposite colors. So this is one of those experiments you just have to kind of do, and it's not super important that you understand the physiology behind it, all right? So that concludes our PowerPoint lecture. Now, what I would do is go through the lab activities this week, and if you have any questions as you go through and answer the questions, send me an email or come talk to me during office hours. All right, everyone, good luck and have fun with this lab.